I will introduce Ariane. She is currently an Alexander von Humboldt Fellow hosted at the Heidelberg Institute for Theoretical Studies in Germany. She obtained her PhD in biochemistry at the University of Sao Paulo in Brazil in 2017. She has been working on the development and application of computer-aided methods, such as chemoinformatics and molecular dynamic simulations, to understand ligand protein binding, especially binding kinetics. In 2020, she was one of the co-organizers of the Latinx Chem, a Twitter conference for chemists from Latin America. So welcome, Ariane, and now you can start.
Thank you for this kind introduction. I'll share my screen. Can you see my screen?
Yes, perfectly.
Okay, wonderful. So hello, everyone. First of all, can you hear me well? Can you see the presentation? Everything fine?
Perfect.
Okay, wonderful. First of all, thank you, the organizers, for putting together a lot of women from Latin America to talk about bioinformatics. I'm super happy to be here with you to talk a little bit about my work. Just a small change. I was a postdoc at the Heidelberg Institute for Theoretical Studies, but now I recently moved to another position. I'm right now a junior group leader at the Technical University of Berlin, but this is since July. So today I'll talk a little bit about the work I have been doing estimating ligand binding kinetics using physics-based and data-based approaches. Okay, so I'll start trying to convince you that binding kinetics is important for drug design and it's nice. And then I'll talk about two of the works I did as a postdoc. First, prediction of binding kinetics using a data-driven method, and then prediction of binding kinetics using a physics-based method, molecular dynamic simulations. So let's get started with the introduction. So we are the computational people here, and there are a lot of ways that computational methods can help in the adventure of drug design. So here we have a very brief timeline of drug development, and computational methods can help in identifying a target, a protein that will be used to design the drug. The computational methods can also be used to identify leads and lead optimization and to optimize the properties by evaluating toxicity and excretion and etc. So what I want to highlight to you today is lead identification and lead optimization. So first of all, if you want to identify a small molecule that binds to a protein target, one of the things we had to do is to decide on a quantitative metrics of what is a good and a bad binder. And that's where comes thermodynamics and kinetics of protein-ligand binding. Well, thermodynamics is usually given by the dissociation constant, Kd, and this applies for equilibrium properties. But today I want to highlight to you two metrics that do not apply to equilibrium systems. One of them is K-on, which is the association rate constant, and the other is K-off, which is the dissociation rate constant or the rate of complex dissociation. So here we have just a very simple scheme of the protein in orange and the ligand in red, and they are binding here in the bound state. So if we think about K-off, sometimes in the literature is also common to refer to the inverse of K-off, the residence time or tau. And the reason why K-on and K-off are so special is because they are better descriptors for non-equilibrium systems. And a very important class of non-equilibrium systems are us and our cells. They are constantly working out of equilibrium. And binding kinetics is also an emergent topic in drug design. So this was first proposed in 2006 in this paper at the Nature Review Drug Discovery. And another thing that highlights the importance of drug design is this association that happened a few years ago, but ended already. And this was a consortium between pharmaceutical industries and universities here in Europe, this K4DD, Kinetics for Drug Discovery. And the idea of this consortium was to advance experimental and computational methods to study binding kinetics for drug design, especially K-off values. And the reason why binding kinetics is so important is that kinetic rates may be better correlated with in vivo efficacy because they describe better non-equilibrium systems. And just to give you an idea, if we have a longer residence time, it means that the ligand spends more time bound to the protein. This leads to longer physiological effects and also less off-target binding. And here we have an example of this from this paper from 2012. So the authors were looking at inhibitors of adenosine A2A receptor, and they quantified the experimental efficacy inside cells. So here in the y-axis, we have the, oh, what's going on? Is everything okay?
Yes, I think so.
Okay, I heard a noise. So here, this is in the y-axis, we have the efficacy of the drug tested inside cells, and here also the efficacy. And here we have the logarithm of the inhibition constant, which is an equilibrium measurement. And here we have the logarithm of the residence time, which is the inverse of K off. And here, if we look at the coefficients of determination, we can see that the coefficients of determination is much higher, closer to 1, when we compare the efficacy with the logarithm of the residence time, which is just an example that kinetic rates are better correlated with in vivo efficacy. Okay, and then if we, the computational people, want to characterize kinetic rates, there are two ways of doing it. We can use physics-based methods, for instance, molecular dynamic simulations, but we usually have to combine MD simulations with enhanced sampling strategies because ligand binding and unbinding happens in a time scale that is beyond the time scale achieved by MD simulations. The advantage of using these methods is that we don't need experimental data for this, but nonetheless, the predictions can take days to happen. We need to wait for the simulations to finish. And on the other hand, we can use data-driven or knowledge-based methods. And these methods are used machine learning methods, regression. And the disadvantage here is that we require experimental data for training our methods. And the larger the data sets we have for training, the better. But once we have the method parameterized, the advantage here is that we can make predictions in seconds or minutes. And if you want to know more about these methods to compute kinetic rates, you can check this website maintained by my former boss, Professor Rebecca Wade. Or for physics-based methods, you can also see this review I published last year. And now I hope you are convinced that binding kinetics is super interesting for drug design. And now we are going to talk a little bit about the work I've done in the last 2 or 3 years. And first of them, prediction of binding kinetics using a data-driven method. So the other thing we'll do in this presentation, we'll do two things, talk about binding kinetics and talk about kinases. So now let's talk about kinases. So kinases are one of the most pursued targets in drug design because of their critical roles in cellular signaling. They are involved in a lot of important things like proliferation, survival, and cell death. And in 2015, the Food and Drug Agency in the US approved 28 kinase inhibitors to be used as drugs. This number is probably even bigger now. For this part of the talk, we'll talk about p38 MAP kinase. And this has pharmaceutical interest because it regulates the production of pro-inflammatory cytokines. So if we design drugs against this kinase, we can control inflammatory processes. In this work, what we wanted to do was to obtain a model to predict K-off rates for inhibitors of MAP kinase, and also develop a method at the same time that accounts for ligand and protein flexibility. And for this part, we use a method called comparative binding energy analysis, also known by this short name, COMBINE analysis. So COMBINE was published first by my former boss, Professor Rebecca Wade, in this paper from 1995. And it's a data-driven method. And the idea is that, first of all, we need a data set to train the method, and then we can make predictions with this afterwards. So first, we look at the interaction energies, V, between the ligand and each protein residue. And these interaction energies are obtained from the structure of the ligand-protein complex. And this usually comes from crystal structures or from docking, for instance. And here we have the interaction energies, and they are composed of the van der Waals interaction energies between the ligand and heavy residue, and the electrostatic interaction energies between the ligand and heavy residue. And once we have these interaction energies, we select among them a few energies, and we scale them by coefficients or weights, W, obtained from partial least square regression. So first we need to have a data set with experimental K-offs available, and then for these protein complexes, we compute these interaction energies, and then we scale the coefficients W and C. And afterwards, once we know W and C, we can use these values to estimate kinetic rates for complexes whose kinetic rates are unknown. So far, so good, but we still don't have the flexibility. So previously in that paper I showed you before, the interaction energies were computed from one structure of the protein-ligand complex. And what we did here was to change the method to now make it possible to compute the interaction energies from multiple structures to describe one single protein-ligand complex. So here we have the logarithm of I, where I is any response variable, in our case, K off. And previously, we computed this log I using just one structure. But now what I did in my work was to change this logarithm of the computed logarithm of I, and now we obtain this by an exponential averaging scheme of n structures used to describe the protein-ligand complex. So if you want to know more about the method, you can check the publication this year at the Journal of Chemical Information and Modeling, or you can also check the code on GitHub. To obtain the complexes to compute the interaction energies V, we use either one crystal structure or 10 structures obtained from ensemble docking. And the ensemble of 10 protein structures to do ensemble docking were obtained from molecular dynamic simulations of 1.2 microseconds of P38 kinase. And that's it. Now we are moving to the results. So our data set was made of 33 inhibitors. So we put 22 of these inhibitors in the training set and 11 of them in the test set. And to choose the optimal number of variables, latent variables, we use leave one out cross validation. So we did two models to compare. We either computed the interaction energies using one single structure, using usually the crystal structure, or we computed the interaction energies using an ensemble of 10 structures to describe each ligand protein complex in the data set. And for this model, the best number of latent variables was 2, and for this model, 1. And here you can see the comparison between the experimental logarithm of K off and the computed logarithm of K off for the model using single structures and the model using the ensemble of 10 structures. So in black, we have the training set, and in pink, the test set. So if we look at the training, the coefficients of determination, the R-square value for the training set, we have here 0.86 and 0.83. Meaning that in both cases, the models could be equally well trained. But then when we look at the test set, we can see here that the test set is the R-square value is 0.79, and here is 0.72. Meaning that for the model using a single structure, the model had a higher predictive power, which was a bit of disappointing because we were expecting that with the incorporation of flexibility, we would have a higher predictive power, but it didn't happen. And then when we do these machine learning methods or chemoinformatics, we don't want only to see a number with the prediction, but we also, at least me as a biochemist, I also want to know why these numbers are the way that they are, or in other words, we also want to get mechanistic insights of what's going on. So here we are making a comparison of BIRB796 bound to kinase and imatinib bound to kinase. So this ligand here has a low K-off value or a large residence time, and that's usually what we want in a drug. And this drug is exactly the opposite. It has a high K-off or a short residence time. And what I'm showing you here in green are key interactions that were identified in combined analysis, which had a high weight, meaning that they are probably important to understand the K-off values. And here, if we compare these two ligands, what we see that characterizes a good drug or a drug with a low K-off value is that we usually have two hydrogen bonds with this glutamate 71, and we usually have hydrophobic interactions with this lysine 53 and hydrophobic interactions in this pocket here made by these three residues, glutamate 71, arginine 67, and arginine 70. And another thing we learned from this model was the role of this lysine here. So first, we are looking at the weights of the electrostatic energies for different residues. And what happened here is that lysine had a negative weight. This is very strange, and I'll explain you why, because this means that whenever the ligand made a strong interaction or a hydrogen bond with lysine, this ligand usually had a short residence time, which is very counterintuitive, because if the ligand has a hydrogen bond with a residue, we usually expect the opposite. The ligand will have a good bind to the binding site, and therefore we'll have a longer residence time. And then we started looking again at the crystal structures, and what we saw is that, in fact, what happens is that when ligands have a long residence time, they do not interact with lysine, and lysine is doing this hydrogen bond with aspartate mediated by a water molecule. So when the ligand is making a hydrogen bond with lysine, this means that this lock, this molecular lock here is not available, and the ligand will go out faster. And this was a very interesting result from this work. So conclusions from this first part, we wanted to develop a method to incorporate flexibility in combined analysis. And we did this by computing interaction energies from multiple structures to represent one protein ligand complex, and later putting together other predictions using an exponential averaging scheme. And then we compared models using single structures or an ensemble of structures to make predictions. And we saw that the model using single structures had a higher predictive power. And finally, what we saw is that combined analysis is able to identify key interactions that contribute to variance in binding kinetics and can be further used to guide drug design for kinases. Now, the third and last part is the prediction of binding kinetics using a physics-based method. Again, we'll talk about kinases, but for this part, we use FAK and PYK2, which had 61% sequence identity with FAK. And these kinases are important because they're involved in cell migration and survival and in cancer. And the pharmaceutical interest of these kinases is tumor proliferation and metastasis. So if we can inhibit these kinases, we can better deal with cancer. And for the second part of the work, we knew of one drug that could bind to both kinases. And this drug had a much higher residence time for FAK than for PYK2. So we teamed up with experimental researchers from Merck and also from the Goethe University in Frankfurt. And we did experimental characterization of a series of inhibitors of kinases. And we wanted to obtain mechanistic insights from experiments and simulations about the kinetic selectivity between FAK and PYK2. Or in other words, why are there drugs that have a long residence time for FAK, but not for PYK2? And if you want to know more about this work, it was also published this year in Cell Chemical Biology. So for this part of the work, we used molecular dynamic simulations. But the challenge of studying unbinding through molecular dynamic simulations is that unbinding usually takes milliseconds or more to happen, while molecular dynamic simulations usually achieve only the microsecond time scale. So to overcome this, we usually have to combine MD simulations with enhanced sampling methods to increase our chances of seeing unbinding events. For this part, we use tau-RANDI. So tau-RANDI means tau random acceleration molecular dynamics. And the idea is that in this method, we facilitate dissociation of the ligand by applying a force of constant magnitude and random orientation in the center of mass of the ligand. And constant magnitude is important because here we don't have absolute K-off values, but we can compute relative K-off values and compare the different ligands to say which one has a longer or shorter residence time. And in this work, we used the version of tau-RANDI developed in 2018 by my former boss, Rebecca Wade, and my former colleague, Daria Koch. And this version was for NAND. Nowadays, there's another version available for GROMACS published last year. So here we have a very brief example of how tau-RANDI works. So first we have to equilibrate the system, and then we'll start applying a force of random orientation, the center of mass of the ligands here in pink. So we apply the force, and then after time delta t, we check the displacement of the ligand, delta x. If this displacement is larger than a threshold, then we keep the direction. But if the displacement is shorter, smaller than a threshold, then we change the direction randomly. And then we keep doing these cycles of propagating the dynamics of the system and then checking the displacement until the ligand is out of the binding site or in the unbound state. And when the ligand reaches the unbound state, this is the relative residence time for this ligand. Now moving for the results. So first thing we did for this work was to characterize the experimental kinetic rates and also the computational kinetic rates using tau-RANDI. So here in red, we have the ligands for FAK, and in green, the inhibitors for PYK2. So here we have a coefficient of determination of 0.91, meaning that tau-RANDI can accurately predict relative residence times. And here we already can see that there are ligands like 10, 1, 11 that have very long residence times for FAK only, but not for PYK2. And that's what we're going to investigate now. What's going on? What is special about these ligands? Okay, what we did was to do intensive data analysis of the simulations, of the equilibrium simulations prior to removing the ligands from the binding site. And here you have the results for this. Here we have inhibitors from 1 to 12, and here we have FAK, and here we have the results for PYK2. In this part, we have the hydrogen bonds also here, but what I want to highlight to you by this black arrow is what's going on with the hydrophobic interactions here and here. So here we can see again the residence time, and we can see that for ligands like 1, 10, and 11, what they have in common is that they have these blue squares, meaning they have frequent interactions with leucine 5, 6, 7. If we look at PYK2, the equivalence leucine is 570, we don't have such interactions for PYK2, only for the inhibitors bound to FAK. So the whole conclusion here is that interactions with leucine 5, 6, 7 leads to longer residence times. And when we check again the crystal structures here, we have FAK bound to ligand 1, a very low K-off value, that's what we want. And here we have PYK2 bound to ligand 1, a high K-off value. And here, if we look at the leucine, here we can see that the DFG motif, an important motif for the function of kinases, this is in a helical shape here. And this is why leucine is interacting strongly with the inhibitor. But here, we don't have this helical motif, and leucine, we don't even see it here in this picture. It should be at some point here. This should be leucine 570, which is not represented here. So the whole conclusion here is that the helical DFG motif present only in FAK leads to strong interactions with leucine 576 and therefore longer residence times. The conclusions for the second part is that a helical DFG motif, as I showed in the last slide, leads to long residence times in FAK, but not in PYK2. And the take-home message here, I hope they convinced you in the introduction that binding kinetics is an emergent topic in drug design because it's better to describe non-equilibrium systems. And computational methods like combine, like tau range can be used to predict kinetic rates and also to reveal key interactions that modulate such kinetic rates. And that's it. I would like to thank my former boss. So this whole work was done at the Heidelberg Institute for Theoretical Studies during my time as a postdoctoral researcher there. And I had the pleasure to work with Professor Rebecca Wade here. And for the first part of the talk, Fabian Ormersbach is also a co-author. He helped me with the code he developed to align ligands. And for the second part, I had the help of Daria Koch, represented here. And also we had the help of experimental collaborators at Merck and Professor Stefan Knapp at the Goethe University in Frankfurt. And of course, I would like to thank all the sponsors, the Klaus Schiller Foundation. Initially, I was sponsored with a postdoctoral fellowship from the Excellent Cluster Cell Networks. And afterwards, I was sponsored by a CAPES Humboldt Fellowship sponsored by the Humboldt Foundation in Germany and CAPES in Brazil. And if you allow me a quick advertisement, I'll have an open PhD position to work in my lab, and this will be advertised soon. This position will be at TU Berlin to study substrate and inhibitor dissociation from hydrogenases. And if you want to know more, just email me. That's it. Thank you for your attention.
Thanks a lot. So now we are open to questions. So I'm checking here in the question and answers side. So okay, Mercedes or Julieta says, the code of this approach for kinases could be adapted for data of another targets in PDB or not that have inhibitors with KI or IC50?
Which code? Oh my God, I
yes,
talked about combine and tau range. I mean, in principle, both could be used. Tau range, the thing is, tau range can be used for whatever protein ligand system you have. It doesn't require training, but combine you can use for whatever you want to predict. But the thing is, you need to have data to train this method. You need to have at least, let's say, 20 or 30 data points to train the method. But you can use it for other things. I used it to predict K-off, but you can use it to predict IC50s or inhibitor constants, Ki.
So Fabiani also asks if you calculate the inhibition constant, the Ki.
No, I didn't. No, sorry, not for the second part of the talk, but for the first part of the talk, I also calculated not Ki, but what was available for it? I don't remember. Sorry, I think it was Ki or Kd. Anyway, I calculated one of them. I think it was Kg for a p38 map kinase. And the thing is that the correlation was worse. So the method somehow for this particular case worked better for K-off values of p38 map kinase. But I know other cases where if you use K-off, Ki, or Kg, they can be equally well predicted. So for this specific case, p38 map kinase, it's in the supporting information of my paper. The results are not so good. So we have a lower coefficient of determination.
Thanks a lot.